Portcullis House.
Can I welcome witnesses to this morning's session? We want to start off by trying to get a better understanding of the level of investment in prion-related technologies and the extent to which this has been taken up by the NHS and so on. So if perhaps we can start off by going down the line and inviting you all to, first of all, introduce yourselves and briefly setting out the prion-related technologies that your organisations have been involved in developing. So we'll start with you.
Good morning. My name is Dr. Kelly Board, and I am a technical specialist for the DuPont chemicals and fluoroproducts businesses, specifically for glycolic acid, oxone, and chlorine dioxide. Prior to this, I worked as a senior research chemist for the disinfectants business, and from 2008 to 2010, I was involved in the Rely on Prion Inactivator project with a technical focus on commercialisation. I joined DuPont in 2005 after having completed my PhD in inorganic chemistry. So the product that we developed was through working with DGen Limited, which is a spin-out company of the MRC Prion units headed by Professor Collinge. And we helped to develop a prion decontamination product for surgical instruments. So the background to this is that our former technical director at DuPont approached Professor Collinge's group after seeing their research demonstrating prion inactivation on surgical instruments using a surfactant and a blend of enzymes. Our company had marketed a high-level disinfectant for surgical instruments called Rely on Perisafe since 1998, and Dr. Crowd saw an opportunity to incorporate this disinfectant technology with that of the MRC Prion unit. The product was launched on the 1st of May in 2007, and I was brought into the project in April 2008, preparing the second submission to the RRP dossier. I was a subsequent point of contact for the rapid review panel and worked on commercialisation efforts until the project was put on hold in 2010. The product itself, Rely on Prion Inactivator, or RPI, it rapidly reduces the potential risk of prion transmission via surgical instruments through use as a manual pre-soak product prior to the usual decontamination methods. It is a powder product comprising 3 packs. Pack 1 is an oxidising disinfectant, Pack 2 is an anionic surfactant, and Pack 3 is a blend of protease enzymes. The product is used by adding each pack sequentially to water at 50 degrees Celsius. The instruments are then fully submerged for a 10-minute contact time, after which time the instruments are then transferred to be sterilised by autoclave or decontaminated by washer disinfectors. The product is at near neutral pH and contains a blend of corrosion inhibitors to preserve the delicate instrument integrity. RPI has been validated by 3 prion methodologies, and it reduces the risk of infection by greater than 1 million-fold.
Okay. Thank you very much. If we can move along.
Hello and good morning. My name is Dr. Stephen Burton. I'm the Chief Executive of a company called ProMetic Biosciences Limited. This is a UK-based subsidiary of a Canadian corporation called ProMetic Biosciences. We have been involved in the development of prion capture and removal technology, sometimes referred to as prion filters, since 2001, when we formed a strategic collaboration, a commercial joint venture, in fact, with the American Red Cross. This was at a time before the first identified transmission of variant CJD by blood, but there was a significant level of concern at the time that that might happen, and that measures to counter this threat should be developed. We undertook a very large program of work involving several universities, the Red Cross, ProMetic, a French medical device manufacturing company called Macapharma. You asked about the investment costs. I can say that between the American Red Cross and ProMetic, to date, we have invested in excess of $50 million in the development of this technology and the applications of this technology. So a very substantial investment. This was made because there was an identified threat through transmission of CJD through blood. Prion filtration, unlike testing, doesn't require any sort of diagnosis. It's a more generic approach to combating the problem. So essentially, if the variant CJD prion is present, it's on passage through this filter. It's a very specific type of filter that has an active surface that's designed to capture the prion protein, and it will capture the prion and remove it. The device, the first product is a medical device called PCAPS. This is a prion removal filter for red cells. This gained CE mark approval in 2006, which means that since 2006 it's been available for use in the processing of blood. It's undergone very extensive testing in several laboratories around the world. It's been shown to be highly effective in capturing and removing prions. It's shown to be very safe. It has undergone numerous trials whereby the filtered blood has been administered to volunteers and to patients, and I think in total there's something like 4 clinical studies been undertaken, and no evidence of any adverse events. So it's a very safe device. The blood that's filtered is very well tolerated. As you might be aware, we have been engaged in discussions with the UK Blood Services for adoption of this device, but it remains at this point in time that there's yet to be a decision in relation to the adoption of that product.
Professor Collinge.
Good morning. I'm Professor of Neurology at UCL Institute of Neurology and at the National Hospital for Neurology and Neurosurgery, and I direct the Medical Research Council's Prion unit, a research unit specifically set up to tackle the problems caused by BSE and variant CJD in 1998 in response to the BSE crisis. We have about 100 scientists and support staff working on understanding prion diseases and finding effective solutions to them. The particular programs that we have in terms of trying to produce solutions for patients are development of a blood test, which we talked about last time I was here, which we published a prototype version of in the Lancet 3 years ago. We developed, at the request of the Department of Health, who funded this work in the unit, means to decontaminate surgical instruments by developing a combination of enzymes and detergents that would effectively destroy prions on metal surfaces. Work that was subsequently licensed, as you heard, to DuPont to turn into a real practical product. And we also have a major activity on developing treatments for prion diseases such as CJD. We have 2 programs that are in an advanced stage. One is to develop classical small molecule drugs, and that's a collaboration with GlaxoSmithKline. And the second is to develop monoclonal antibodies to treat the disease, which has been taken forward by the MRC unit.
Raeber.
Well, good morning. My name is Alex Raeber. I'm heading research and development at the Prionics company, which is a veterinary diagnostic company headquartered in Switzerland and marketing products for animal health worldwide. To give you a little bit of the background, Prionics was founded in 1997. We were founded with the first commercial product for BSE, which was gaining market access around 2000, 2001. And this was the Prionics Check Western blot, which was validated by the European Commission in 1999. And subsequently it was approved for use in the BSC surveillance and the eradication program in the European Union, and it was listed in Annex 10 of Regulation 999/2001. Subsequently, Prionics has developed further tests for BSC, an ELISA test, which gained approval in 2003, and a lateral flow, a strip test, in 2004. And Prionics is currently still one of the major players in the animal diagnostic for BSC. In addition to those development programs, Prionics started back in 2002 with the development of technologies for prion detection in humans. And in particular, we published seminal work in Nature back in 1997, where we showed that a prion-specific antibody exclusively can capture abnormal forms of the prion protein. And this antibody was then developed into the first prototype of a human blood test. In subsequent years, that was between 2007 and 2011, we evaluated the test together with NIBSC, and the test was later on found not to be sensitive enough and was not further developed into a commercial product. Prionics has also developed further technologies in collaboration with the NIH in the US, a technology which is known as the eQuick technology, and which is probably going to be discussed here a little bit later. Thank you.
Good morning. My name is Nigel Tallboys. I'm responsible for the blood safety business for Terumo BCT, and I also have responsibility in Europe for public affairs and public policy. Terumo BCT is a subsidiary of a Japanese medical technology company called Terumo, and we are the global leader in blood component and cellular technologies. It is the only company with a unique combination of various collection, processing, and blood safety technologies. We, in fact, do not manufacture a specific prion reduction or prion testing technology. We have a broader pathogen reduction technology. This technology, which is known as Mirasol, was launched in Europe in 2007 and reduces broad spectrum of pathogens such as viruses, bacteria, and parasites. This technology has had hundreds of millions of dollars invested in it, and is available and used in approximately 18 countries, primarily in Europe, but also in Russia and in North Africa. And approximately 60 different centers are using this technology. We've worked not only in Europe but we also have a very large program of development with the Department of Defense in the USA, in which we're developing a system for battlefield applications of this technology that can be used in situations where there is with large trauma and safe blood needs to be transfused. Thank you.
Thank you. In different ways, some of you have partially answered some of my follow-up questions, but I'll start with you, Dr. Burton, because you were the most complete in what I was going to say next. I'm trying to get a handle on approximately how much has been invested in these technologies. Well, you've given us a figure already in your case. How much financial support you've had from the government, either directly or through the research councils. And I'm also interested, as an aside, for those of you with broader international connections, whether there are any similar arrangements that have benefited your research programs from other governments. To what extent have these products generated a financial return? And of course, the key one in terms of the UK, how much are being used within the NHS and the UK Blood Service. So, as you started that theme, Dr. Burton, I'll start with you.
Okay. The support that we've had for the development of this product has really come from outside the UK.
From?
Outside the UK. To my knowledge, we've not had any sort of grant support or other supporting kind in relation to the development of this technology. We were fortunate to form this collaboration with the American Red Cross. This, as you know, is a part governmental, part voluntary organisation in the US. They were concerned globally about the issue of variant CJD transmission by blood, and were prepared to invest money in the development of a product to combat that threat. There has not been a similar arrangement in the UK. The UK government and the Department of Health has, I would say, relied upon industry to pick up the brunt of the cost of developing a product in the hope that it will at some point be used. Of course, everybody, I guess, in a way hopes it won't be used and that this disease is not something that is a thing of the past. But unfortunately, I don't think we can say that right now, and these measures are required. So in our case, we had, I would say, little if any support from the UK for the development of this technology.
But you've now got CE approval. Are you selling the product in the UK?
We are not. There was a small number of filters were supplied to the UK Blood Services as a part of the PRISM clinical trial. But other than that, there is no current supply or sale of product in the UK. For the PCAP filter, there is another application of the Prion technology that we have developed, which relates to plasma as opposed to red cells. We have established a collaboration with a Swiss company called Optopharma. They have a virally inactivated prion-reduced plasma called Optoplas. This is an approved product in the UK, elsewhere in Europe and in the US, and there are growing sales of that plasma product, and the technology that we developed is being used in relation to the manufacture of that plasma product, but currently it's not being used in the UK in relation to red cell processing.
And so in the UK, I guess, it certainly hasn't produced a financial return yet?
No.
Elsewhere?
Elsewhere, we have a small return, I would say, in relation to the investment that we've made from the plasma business. That is growing because it is a safer form of plasma compared to fresh frozen plasma, for example. So we ultimately may see a return on our investment. But the focal point really for this work has been red cell filtration. And of course, in the UK, that is determined by the Department of Health and the National Blood Service.
Who's going to go next? Dr. Raeber.
Thank you. Prionics, between 2002 and 2014, when we developed prototype tests for variant CJD, we spent between 5 and 10 million euro for this research program. This money was basically all spent on Prionics' own behalf, so we didn't have any government support, neither from the UK government nor from any other governments. So basically it was invested from our revenues, which were coming from our BSC test and other animal diagnostic tests. We had some funding under the EU Framework Programme 7, a project which was called Priority, which started in 2009 and will end in September 2014. And under this program, Prionics is still involved in the development of a blood test for variant CJD and received about 300,000 euros. with regard to commercial returns out of this program, Prionics didn't have any returns from this research program. Prionics participated 2 times in published tenders by NHSBT. One tender was published in 2007, the other one in 2009. And those were both commercial tenders. Prionics won the 2009 tender for the provision of devices and testing service to the NHSBT. This led to a framework contract with the NHSBT, and under this framework contract, Prionics was basically commissioned to deliver any products if the NHSBT would ask for such products. This was never the case. Prionics never delivered any products to the NHSBT. I might have to add here that the test was during that time of the tender process undergoing evaluation. So this was a parallel process led by the NIBSC, and it turned out that the test would not be fit for purpose, what the NHSBT was asking for.
The majority of our funding has come from internal sources. We've In our technology, the Miracell technology, we have invested in excess of $100 million. There has been some funding from the Department of Defense in the USA, which is approximately $15 million so far. And in addition, there is a co-funding program we have with the Department of Defense, which is about $15 million additional funding from us and $15 million from the Department of Defense. From return standpoint, the product is commercially available and used within the European Union. It's also used in East Europe, North Africa, and in Russia today. And we have some small evaluations starting in Asia. As far as the UK is concerned, there is a SABTO working party that is currently looking at pathogen reduction technologies, and we are working with the SABTO working group on this and providing information to the SABTO working party. But as far as any commercial returns in the UK so far, there are zero.
Dr. Board.
We did not receive any financial support in the forms of grants from the UK or from other countries. DuPont made an investment of around £150,000 in terms of out-of-pocket expenses, excluding the staffing costs that were associated with the project. We have not yet had a financial return. There have been no sales of the product at all, so the product was not taken up by the NHS. We did submit the product for review by the Rapid Review Panel on 2 occasions in 2007 and 2008 in the hope that we would achieve a recommendation 1, because this would have seen the product taken forward for trials in showcase hospitals around the country. However, we obtained recommendations 3 and then subsequently a recommendation 2. The recommendation 2 essentially means that the basic R&D has been completed, and they recognise the product may have potential value, but more in-use evaluations and trials are needed in a healthcare setting. They had additional comments around the fact that use of a manual soak might not be justifiable given the uncertainty surrounding the prevalence of CJD in the population. We had some further contact with the NHS through the Infection Prevention Product Specialist team. They reviewed the evidence that we'd submitted to the Rapid Review Panel, and their further comments were that it would be unlikely that there'd be widespread uptake of novel prion technologies unless they're recommended in authoritative national guidance.
David.
Good morning. I'd like to ask you whether or not you think the main barriers to developing a screening test for variant CJD are commercial or scientific, and if they're commercial, what are the key obstacles? Dr. Raeber, you look as if you're first out of the stall.
Well, there are, of course, a number of commercial barriers. the biggest barrier in order to get a test onto the market is probably the very restricted market. Currently, the main markets are considered to be the UK and France. Those two countries had most of the variant CJD cases, and those two countries would be obviously the first market to go into. Those markets are relatively restricted in terms of blood donations. We're talking about 5 million blood donations, 2.5 in France, 2.5 in the UK. So this is a niche market, and any big company will probably not go into that market. Then probably the second biggest challenge is the regulatory framework. Variant CJD blood tests were put under a new regulatory directive, under the IVD directive, which is the Directive 98/79. And in 2011, they were added to List A of Annex 2, which basically contains blood tests such as HIV and hepatitis, which are the highest regulated group of any diagnostic tests. And this, of course, poses significant investments, costs, and it is considered also a significant barrier. In addition, there are scientific barriers, which means there's still a lot of uncertainty around prions. What is a prion? When is it infectious? And last but not least, I would also say that there are some political barriers. That's why we're all sitting here today. So the market is very much politically influenced, and a company developing a test to a certain requirement, and the requirements are laid down in the common technical specification of the directive, would actually expect to get market authorization for such a test. For this class of test, it is different because there's also a political dimension involved.
I can talk about the broader aspects of blood safety rather than specifically on Prion technologies, but I think it has a lot of similarities. The investment on these technologies is rather large, and it's a continuing investment in the technologies. We have, for example, currently clinical studies that are continuing throughout Europe. We have in the Netherlands, in Italy, in Norway, studies as well as in Canada, and in fact this week our technology is starting a clinical study in Ghana for having a look at the reduction of the transmission of malaria through blood. And although there has been a significant amount of information in the different directives to suggest that these technologies should be used, such as the Blood Directive of 2002, which said that these type of technologies should be used either testing for blood safety or the inactivation of blood products. The uptake has been rather slow, and again, I would like to say that I think there tends to be a lot of political agenda on the uptake of these technologies throughout Europe, not just in the UK. And it was even in 2008 there was the midterm report from the European Environment and Health Action Plan in September 2008 again reiterated that these blood safety technologies for testing and inactivation of blood products should be taken up if available.
Dr. Raeber, you've talked about the market, the main market being the UK and France. What about the United States then? And are there any other significant markets, or is it really that much of a niche market? And what would link to our old ally or foe, if you prefer France?
Well, I mean, the United States, of course, will be a very interesting market, and the question will be if the UK, the blood transfusion service in the UK would start implementing a variant CJD blood test. What kind of an impact would this have on the overall blood supply worldwide, and would other countries need to be followed in order to, adopt the same standard? to me, this is still a question I cannot answer. It's not my expertise. You could, of course, argue if the whole developed world would follow to implement this testing, then the market would be more significant.
Is that where you mentioned politics, the politics of health? Is that where politics comes in?
Excuse me.
Politics. You mentioned earlier on politics.
Right.
The political issues around this. Is that partly a political matter then? Government commitment. Mr. Talboys, you said
probably. I mean, it's probably all about whether. I mean, coming back to the US, would they really see the same safety issue as in the UK?
Right.
And this again would be a political process.
Do you disagree with that?
No, I fully agree. I think that decisions have to be made in healthcare, and whether it's a testing or an inactivation of a particular pathogen or within blood, it has a cost associated with that, and therefore has a cost to the healthcare system, and therefore someone has to pay for this, whether it's in Europe or in the US.
It's more money than policy. It's money in the end. It comes down to hard cash.
Yes.
Dr. Burton.
Just to pick up on that. I would say one needs to look back over quite a lengthy period of time here, because obviously the political situation changes depending upon public sentiment and the perceived risk and need to do something. If one looks back at 2001, 2002, we were recovering from the BSE crisis. We were seeing, we had just seen a very large number, a relatively large number of cases of variant CJD from the oral administration. I'd say a large number. I mean, this is relative. We're talking numbers maybe 25 per annum, deaths per annum at that point at its peak. And then those numbers declined. And I think there was a perception at the time that this is going away, the problem's gone. But that doesn't take into account many other factors, such as the whole genotype issue and the length of the incubation period and things of that nature. So I would say political sentiment started to change around about 2004 to 2006, that maybe this problem was going away. And then, of course, later on we ran into a global financial crisis that put pressures on budgets. There was a document published by the Department of Health, the 5-year strategic plan for NHSBT, where clearly reducing the price of blood is seen as an important objective. And it's actually commented upon in that report that, of course, if additional measures such as Prion filtration or testing had to be implemented, that would have a negative impact in terms of reducing the cost of blood. So I think that there is a political dimension to this.
Right, thank you very much. Just moving on now. Could any of the tests under development be used to detect other pathogens, please? I mean, for example, classical forms of CJD or other currently uncharacterized prion diseases.
I mean, I'm not sure whether I understood the question. So whether this class of tests we were developing for variant CJD, whether this test could also detect other...
Could they detect other pathogens? I mean, is there a wider... If all these tests are being developed, is there actually a wider market out there, I suppose, to use a commercial term? Are there other opportunities to deploy the knowledge?
Well, the knowledge on prion technology, yes, there would be a broader use, but when you develop a test for a specific analyte, and in this case it is the analyte is for variant CJD, the test would be specifically developed for variant CJD, and it could not be used for any other.
I understand that, but you just said that there were other opportunities. Where would they be? Where do you think these tests could be deployed elsewhere? What other medical issues could they be? Could these tests help resolve or help influence?
Well, there are other applications, for example, for classical forms of CJD, sporadic forms of CJD, and there are research programs going on, for example, to discriminate between Alzheimer's disease and Creutzfeldt-Jakob disease.
So Alzheimer's would be a candidate,
right?
Is anybody else going to thoughts on that?
Well, I mean, the tests we're developing are very specifically oriented towards prion detection.
Which detection?
Prion detection,
right.
the infectious agents that cause CJD and variant CJD. So they're highly specific for that indication. Although the test that we've developed is beginning to pick up some cases of classical CJD as well as variant CJD. It's possible it could be adapted for that also.
But I think you said to us before that your line of research has helped us understand other neurological conditions. Is that fair?
That's right. I mean, the fundamental process at a molecular level that's going on in these diseases is about protein aggregation, proteins in the body changing their shape and sticking to each other, forming these seeds which can propagate. And similar processes are happening in Alzheimer's disease and Parkinson's disease, for example. So the science we've done at the MRC unit to develop the blood test, which involves capturing these abnormal proteins on metal powder, could in principle be used for some of these other proteins as well. It's certainly something we're thinking about, whether the same sort of technology could be used to develop blood tests for Alzheimer's disease. But that research is in its infancy at the moment.
Okay, Professor Collinge, I have one last question really for Professor Collinge to start with. Anyway, that's about the scale of the problem. In our last evidence session, Christine Lord suggested that the prevalence of sporadic CJD had increased in recent years from approximately 1 in a million to 1 in 33,000. Does this fit with your understanding? If so, what might this increase be attributed to?
I think there's some misunderstanding there. I mean, the apparent incidence of sporadic CJD is increasing. It's about 100 new cases a year at the moment. That's gone up substantially from the start of surveillance. I mean, that's thought principally to reflect better diagnosis of the disease. That may not be all the explanation. I think it's possible, and we can talk about that some of that may be BSE related. However, I think the specific confusion there is people talk about sporadic CJD occurring at 1 per million. That's not your individual risk. Your risk is 1 per million every year. So 1 per million of the population, it's actually nearer 2 per million a year of the population will develop sporadic CJD. But your lifetime risk of developing sporadic CJD is about 1 in 30,000. So that hasn't really changed. So when people talk about 1 per million, often people interpret that as thinking, oh, it's incredibly rare. I've got a 1 in a million chance of developing this disease. You haven't. You have about a 1 in 30,000 chance of developing it.
Thank you.
Steve.
Thank you. It was Professor Collinge that I just want to concentrate 2 or 3 questions on the MRC unit's Prion test. Could you just tell us what the current status is of the test?
So the prototype test we published in the Lancet 3 years ago, showing that we could pick up 71% of definite cases of variant CJD. Sensitivity of the test, we don't really know what that means. Obviously, we'd like it to be in the 90s. Of course, we don't know whether everybody that has variant CJD has prions in their blood at all. And indeed, we recently published a case history in the Journal of the American Medical Association of a definite variant CJD case, which had virtually no prions detectable in the periphery in the particular system, and we couldn't detect it in the blood. So this would have been a case that would have been missed, for example, by the recent prevalence screening that's been done by Public Health England. So we achieved 71% sensitivity, but that we don't know quite what that means, how much it's possible to increase that. I should say that the test that we developed works on 8 microliters of blood, a tiny amount of blood. A typical blood sample you take from a patient's arm would be more like 8 mils, 1,000 times more than that. So there's plenty of potential to increase the sensitivity of the test by putting larger volumes of analyte into the test. That's not something we've done yet. The important thing in terms of developing a test either to do what we want to do next, which is to conduct a preliminary prevalence screen in the population, or indeed to ultimately produce a test that the National Blood Service could use in a practical sense to screen blood. The key issue is the false positive rate, which needs to be extremely low if you're going to use that for population screening. It also needs to be low in terms of doing a prevalence study to make the numbers reasonable. So we screened 5,000 American samples kindly provided by the American Red Cross of normal US donors. The US population has had minimal exposure to primary exposure to BSE, and therefore we wouldn't have expected to see any true positives in 5,000 Americans, and indeed we didn't. So the test was 100% specific there. So that was very encouraging, and that was our criterion to progress, we hope, to the next stage, and the next stage of evaluation of this prototype that we want to do is to screen 20,000 UK individuals, 20,000 normal UK individuals, and compare that with 20,000 US donors. The purpose of this is twofold. As you know, the Health Protection Agency, now Public Health England, found approximately 1 in 2,000 appendix samples were positive. We did power calculations and worked out that looking at 20,000, comparing 20,000 Americans and 20,000 British samples, we've got a good chance, an adequate chance of showing a significant difference between those two populations in terms of the number of positives. If we do that, we would expect to find 7 positives in the British population and none in the Americans. If that's what we found, then that we could conclude 2 things immediately. One is that our test is capable of detecting carriers, which we don't formally know yet. We've simply looked at patients. And secondly, we would have confirmed that there is indeed a problem in the British donor pool. And in our view, that piece of research is required to make the case to progress that test further. Our test is a laboratory prototype. It's not something that could be used practically at the moment by the blood service, but there's no reason technically why it couldn't be developed in that way. So that we see as the next step, and the discussions we've had with diagnostics companies is very much that they want to see those data first before thinking about whether they'd help us taking it any further. As you've heard from others here, there are other issues as well about whether the market is really worthwhile for companies to progress in, and to what extent the Department of Health and others in government may need to drive that and to provide leadership for that to actually happen, to get over these other barriers.
Okay, so it sounds like you've fallen sort of the valley of death at the moment, haven't you?
That's right.
MRC turned down your funding for that bigger study last year. What are you moving forward at all now? Are you sort of in abeyance in terms of the project, or are you actually continuing without doing that study?
Yes, we were rather set back by that. I think the view of the panel that reviewed that was that we needed to increase the sensitivity of the test further in the first instance.
Would you agree with that?
No, I don't agree with that, and that's not the view of our statistical advisors. The sensitivity is perfectly adequate to do the study that we propose to do. Clearly, if you're going to go on and develop it as a blood screening test, you may want to increase that sensitivity. But as I mentioned earlier, we don't know whether that's possible. It could be that only 70% of people with variant CJD have prions in their blood, and we're picking up all of them. There's an area of uncertainty here. But there's certainly plenty of capacity, as I've mentioned, not least because of the volume of blood we're actually putting into the test is tiny, so there's plenty of capacity to increase that. But what we're now talking about is test development work, which is not what my unit is expert in. We've done the basic science, we've solved the scientific problems, which were formidable to do this. But I'm reluctant for the unit to we don't have the expertise, I'm reluctant for the unit to get drawn into doing a huge amount of routine test development work, because I think that's really a matter for others. What we have a lot of discussions in the unit about this, since we sort of couldn't progress with this, and we have been doing some further work in the unit to see whether we can partially automate the test, because the main cost of screening the 20,000 UK and 20,000 US samples is simply the technician time. It's quite a laborious test at the moment, and so you have to employ probably a dozen technicians to do this in a reasonable time. That's where most of the money goes. So we have robotic systems in the unit that are used for other purposes, and so we're trying to see if we can adapt it to that. The pure purpose for this is to see if we can reduce the cost of doing it, so we can find some way of doing it at reduced cost. But I do feel this is all rather bizarre since my unit was set up to do this, and given the public health considerations that we're having to try and find workarounds to do what actually is a fairly modest piece of work. As I mentioned last time, the funding required to do that we thought was about £750,000. In the scale of what you've been hearing, it's really essentially trivial.
I've got a bit of a contradiction in my text here. So could you just clarify something for me? We've been told that Public Health England suggested that the case for conducting the study would be further increased if it could be shown that prion deposition in the appendix did not predate the BSE crisis. And yet elsewhere in my text it says that it would be strengthened if ineffectivity could be shown to predate BSE. Could you clarify which it is?
I'm not quite sure I understand your question there. You're talking now about the appendix prevalence studies that Public Health England have been doing. Because I think they are now doing a further study on appendices removed from samples prior to 1980. The purpose of that, I think, is to look at appendices from individuals who wouldn't have been exposed to BSE, would have less exposure to BSE, to compare that with the positives they've found so far. Although I must say I'm slightly nervous about that myself, because we don't really know when BSE exposure of the population started. BSE was first recognised in cattle in the UK around 1985. The average incubation period in cattle at the peak of the epidemic was about 5 years. But the range of incubation periods in cattle, like in other species, is very broad. As you reduce the infective dose you give to an animal towards the minimum infective dose of a single infectious unit, the incubation periods increase. And the longest incubation periods you see in cattle, the former Ministry of Agriculture did this experiment in cattle, infecting them with progressively smaller doses. And the maximum incubation periods approach 20 years. So there's likely to have been some exposure of the UK population to BSE well before 1980 also. So that's a caveat in interpreting those studies.
Okay. When we spoke to you in November, you said you'd recently spoken to the Public Health Minister, Jane Ellison. Did you get any feedback, any response from that meeting?
I haven't heard any further from her office now.
Okay. And then lastly, just opening it up, Professor Collinge has been advised to go out to industry to get financial support. From your company's perspectives, is this a type of technology that you would invest in at this stage of development, and if not, why not? And I'm not asking you to get out your checkbook here. Why not?
Well, I think it's, as Professor Collinge mentioned, it's probably too premature to make a decision whether a company such as ours would invest in this technology, because so far it's a research-based test. There has been a couple of papers published. One of the main concerns from our side is that there hasn't been an independent body involved in validating that test, for example, such as the NIBSC, which has been involved in validating tests in the UK from probably more than half a dozen companies that were active in test development between 2007 and 2011. And this is a very important part for a commercial company to get an independent view on a test development. So I think currently it's too early. And as Professor John Collinge mentioned, it's probably important that further evaluations are being carried out, either on behalf of the NIBSC or NHSBT, and the results of those evaluations will show whether there is an interest from industry.
And is that a yes, or would you agree with that, essentially?
Yes. I mean, I think based on our experience, which is not testing, but it's Prion filtration as a different measure in relation to VCJD. One would have, if I was asked the question today, would we invest another 50 million to develop a technology in this area? I think our answer would be no, unless there is a very clear undertaking that if we developed a product and that product met the target objectives and specifications, that it would be used and there would indeed be a market for that product. companies are not going to invest in the development of products if they're not sure in the knowledge that there's a market for that product.
And the others agree.
Thank you, Chair. Dr. Raeber, I understand that the NHS blood and transplant had rejected the blood test that you were developing back in 2009, saying that it was unsuitable. Could you tell us what explanation was given to Prionics about why that test was unsuitable?
Yes, so maybe I have to go a little bit more into what test development really means within the framework of the NIBSC process. Because there's very limited samples from variant CJD available for test development. That's probably one of the main restrictions for test developing, and that's why basically the whole Prion community has agreed on using so-called spiked samples. Spiked samples are samples with human plasma where variant CJD brain is spiked in limiting dilutions into human plasma. And those samples are then being used sort of to mimic a naturally infected variant CJD plasma. So we have been using such samples for basically most of the development work, and the NIBSC provides for a first step such spiked samples to test developers. In a second step, there are animal samples being used because basically the argument is, the rationale behind it, that an animal with prion disease has similar infectivity in the blood as a human. So this is probably the second best choice of samples because there's no human variant CJD samples available. And then in the third stage, negative human plasma samples are being screened, large numbers, a couple of thousand. And those three stages were all passed successfully by our test, which was the Prionics check variant CJD test. So then the last stage was getting access to those very rare variant CJD plasma samples. And in our testing evaluation, the NIBSC provided us with 2 variant CJD plasma samples. We tested those 2 samples. They were blinded within a panel of negative plasma samples, and it turned out that they were just very rarely on the cutoff. So basically, the decision was that the test was not sufficiently fit for purpose. Now, from a test developer perspective, of course, you would say this is really not adequate to make a decision based on 2 samples. If you're looking at the test from the MRC, it had a 70% sensitivity on variant CJD plasma samples, which means there are also variant CJD plasma samples which were missed by that test. So let's say we maybe got access to 2 samples which were really negative, then this final evaluation stage wouldn't really say anything about the test performance on real variant CJD samples.
Okay, so you don't think this was a fair decision?
Excuse me?
You don't think this was a fair decision from NHS?
I think it was not fair in as much that based on 2 samples the whole test was abandoned. I think the real issue lies in the process how to get access to such samples. As a foreign company, we really had big challenges to get access to such samples.
Is there an alternative process that NHS blood and transplant could undertake when examining new tests that are available?
I didn't.
You mentioned that it would be better if there was more samples available, but other than that, would there be an alternative process that NHS blood and transplant could undertake in order to examine the success of new tests?
I don't think it's probably a reason of the process which is not adequate. I think maybe NIBSC was not having access to enough samples. That's why they could only give this very limited amount of samples to company. But I remember in 2011 there were just 2 companies left who were reaching this final stage of development, and both companies received 2 samples. So I was just wondering whether are there not more samples. We knew there were more samples available, but those samples would not be made available to test manufacturers.
Okay. Since 2009, when this decision was made, has Prionics developed this test any further, or is development stalled?
No, I mean, Prionics did continue test development, so we decided to move on to another technology, which was, getting to a stage where it looked really interesting at that time. This is a so-called amplification technology. It's also known as EQUIC, for Enhanced Quaking Induced Conversion Assay. It's an amplification technology which allows to amplify very, very small quantities of prions in blood. And Prionics had collaborated in the last couple of years with the NIH in the US, and we have published in 2011 a paper where we showed that this test would detect up to attogram of prions in blood. So from a sensitivity point of view, this test looks very, very promising. Currently, the NHS-BT is in the process of evaluating that test, and Prionics is still involved in that test, basically from a intellectual property point of view.
Okay. Has there been any other conversations with the UK government following the early success of this test to develop it?
No.
Okay, thank you.
Sarah.
Yeah, thank you very much. I'd like to really carry on exploring the access to CJD samples as an issue in preventing blood tests from developing. And if, with the permission of the Chair, unusually in our deliberations we look at the impact on real people. Our conversation is very much, as it has been this morning, around institutions and processes. We had very powerful evidence from Christine Lloyd, the mother of a victim, her son Andrew Black, and she repeatedly criticised this issue of lack of access to samples that she and other victims feel should be made available to help find tests. And so I think it's important that we actually think there is a human side to everything that we're talking about this morning. So I'd really like to pick up on, do you agree that there should be limitations on the access to rare samples? And if you do, how should they be controlled? Perhaps Dr. Raeber could answer that question first.
I think in principle, the NIBSC, I think, has done an excellent job on setting up the process, and I think I really have to credit the NIBSC for doing that. In my opinion, I think the NIBSC process should probably have been applicable to all test developers, because as a foreign manufacturer, we had the feeling that we were not sort of treated the same way as maybe other stakeholders, and I think it's not the process; it's more probably the kind of decisions that were made at what stage a company would get access to those rare samples.
Would anybody else like to comment?
Yes, the background to this, of course, is that CJD is thankfully a rare disease, and there aren't many blood samples. Many of them are very small, and what was happening with respect to the two units in the UK that see most of the patients and take samples for research purposes, which is my unit in London and the surveillance unit in Edinburgh, there were requests, quite unreasonable requests for large amounts of samples from often very small companies with tests that were not very plausible. And had these been agreed to, the whole lot of samples would have disappeared, and no one would have been able to work on this disease. And that was what's led to the setting up this CJD Resource Centre Committee, which has a defined set of samples and a defined process and algorithm to deciding who gets access to these samples. And that access is based on test performance on less valuable samples, like the spiked samples Dr. Raeber has described. My unit and also colleagues in Edinburgh were asked by that centre to provide samples for that collection, which we've done. So I think that process is very important, because it's just not, with all due respect to commercial colleagues here, often claims are made about tests that don't really have any validity. As academics, we publish our work in peer-reviewed journals for everyone to look at. That's not usually the case for commercial developers, and you often don't know quite what's really going on, how the tests work. And I think it's important that these very valuable samples are only made available if there's really a scientific case, if it's plausible that the test is going to work. Otherwise, we just lose all the samples and we're in serious trouble. So that's why that process was introduced to act as a gatekeeper to access these very valuable samples. I should say, with respect to Christine Lord's comments, which I read in the transcript, I know Mrs. Lord very well. There is an issue. I should say we've had, my units had direct requests from companies for samples, which has put us in a difficult position because we as the MRC are signed up to this national process in terms of access to samples. A Canadian company, for example, that claimed to be very close to having a test asked us for samples, and in fact had been in discussion with patients' relatives to see whether samples could be obtained. And we tried to help. In fact, I asked the chair of the committee after discussing it with MRC head office, look, can I provide some samples outside this arrangement? And the committee agreed, so we provided some samples directly to that Canadian company. And also we've been providing samples recently to a French group that's working on a blood test. So it puts me in a slightly uncomfortable position because you want to help, but there is this national process which really we should stick to. Otherwise, I think it's less of a problem now. In the early days, there were lots of small companies claiming to have tests, and they all wanted bucket loads of samples. It was just impossible. So I think there had to be a control over this. And I think NIBs are doing as good a job as can be done to try and control access to the samples.
Yeah, I'm sure we'd all agree that there has to be some sort of process because there is a limited amount of samples. Yeah. But I'd be interested to see the commercial colleagues here, how they do respond to the comments that you've made. And if anybody could think a way in which the process could be improved, because obviously we received considerable evidence that the process is in fact impeding development of tests. So Dr. Raeber, perhaps if you'd like to respond.
If I could just say one more thing. We published in The Lancet, again it's a question of claims and reality, and a lot of the data of course is not in the public domain in the way academics publish data. What we did publish in our Lancet paper is that which provided really a benchmark of where you need to be with test sensitivity. Spike samples are very important to work with first of all because the real samples are so valuable. The test that we developed was able to detect spike samples brain diluted 10,000 million times, 10 to the order 10. And that shows the ballpark tests need to be in to realistically be detecting in human blood. And when we published that was 5 orders of magnitude above any other published test. And so I think it is important that there's some scientific view that this test is in the right ballpark to begin with spike samples, because if it's not, we're simply wasting the real samples.
Well, how could the process be improved, your question? I think the process as it is, as I have mentioned, is fine. I think there's nothing that is wrong with the process. I think it's more that the process should be adhered to more strictly. So basically all the samples should be made available to the NIBSC, and the samples should then be really given to those tests that show the biggest promise. And I think that's important because you can only compare tests when they have been evaluated on the same samples. And that was, as I mentioned before, done in a very meticulous way by the NIBSC. When you follow that process, then you really have a high chance of giving access to those samples to those test developers who have the best test.
Okay. Would anybody else like to comment? Perhaps Dr. Kelly.
Well, our product was developed in collaboration with DGen, so we weren't involved with the Prion testing part from our side, so that work was completed by Professor Collinge's group.
Can I just make one other technical point which might be relevant here, and that is, in our experience, it has been an issue when we've been asked for access to samples, is different test developers often need different analytes. For example, they want the blood sample taken in different ways with different anticoagulants and so on, and it can be quite complicated. That was a complication with respect to one request that I think you may have heard of with respect to patients wanting samples sent. to a test manufacturer. The samples simply weren't suitable for that purpose. The manufacturer couldn't use them. And so, it's a little more complicated than it might seem.
Yeah.
They're not just samples that can be used for any purpose.
No, I understand. Just one final question, perhaps for Professor Collinge. Our committee has been very concerned, as you say, about commercial developers not making their research findings available, and share the concerns that you've raised. However, it's our understanding that various protocols are being developed so that private developers, especially in the pharmaceutical industry, are actually agreeing to make their research findings available. If such protocols could be agreed with developers who come to you asking for samples, would you be more willing to work with them, understanding that the results of their research would find their way into public domain?
We're happy to work with anyone that will help produce a blood test for this disease, but I think, as I said, it's important as a national process here, rather than putting individual investigators under pressure to provide samples. I think there needs to be an officially sanctioned process with criteria and an independent committee looking at it, so that you have transparency about this. But I think it would help if the data were actually publicly available. Some data are made available to these committees, but it's all confidential. It's hard to know the quality of what's the substance of claims that are being made.
Okay, thank you.
Do you have to go through the NIBSC process yourself when your own researchers want to utilise the samples?
No, we don't, and that was agreed up front. I mean, we're a medical research council unit that are collecting these samples for a ethically determined research program, so it would be a bit crazy creating a separate process for ourselves.
David?
But we supply all samples to NIBS that they've requested.
I'm afraid I can't remember whether it was Dr. Raeber or Professor Collinge who said that there was a request from a French company. Does France not provide samples? I mean, they have the second highest incidence.
It wasn't a French company, it was a group of independent French academic researchers.
The question still arises, are the French using the same sort of protocols? Are they? Is there a common protocol, or are they simply not providing samples?
Well, I mean, the French have variant CJD. In fact, in recent years they've had more than the UK. I can't speak for the French regulations as to how easy it is to access samples in France.
Yeah.
Graham?
I mean, perhaps does Dr. Raeber know? I mean, because he operates in France as well.
I don't know whether the French really followed the UK process. That was your question, right?
Well, or a similar process.
Or whether they have
a process of their own.
I wouldn't know of a process which is similar to the NIBSC process in France. I just know that many companies, that were involved in test developing were actually coming from abroad. So there were Canadian companies, and they all followed the NIBSC process. So the NIBSC process was very closely linked to the common technical specification in the IVD directive. So it really formed a good basis for test developers. So we knew what were the requirements later also to get the test approved. So I think the common technical specification laid down in the EU directive and the NIBSC process, those really matched. And those made it a very good process to follow.
Graham, thank you.
Last time you were here, you said you believed that the transmission of variant CJD via surgical instruments was a significant problem. If that is the case, why have there been no cases reported where variant CJD has been transmitted in that particular
well, we don't. What I was trying to say is that there's clearly a risk there. We haven't been able to quantify that risk. It's correct that we don't know of any cases of secondary variant CJD that can be attributed to surgical instruments at this point in time. But it can't be excluded that some of the cases we've seen so far of variant CJD have been related to that. It's a question of how you would tell. That really comes from epidemiological studies. And since a history of prior surgery, particularly a history of prior dental surgery, is so common, many patients you see will have had prior surgery, and certainly virtually all of them will have had dentistry. So it's hard to make that link. The link has been made with sporadic CJD, where there have been clearly documented instances of transmission of Prions by surgical instruments, by neurosurgical instruments. And there's also epidemiological evidence from several countries now that patients developing classical CJD are more likely to have had abdominal surgery beforehand, for example. So there are sufficient numbers that an epidemiological link has been shown. But you're right, we don't have definite evidence that a surgically transmitted case of variant CJD has occurred, but that's perhaps not surprising at this point in time. What we do have is a great deal of scientific evidence that, first of all, that the infective agent in variant CJD is much more widely distributed in the body than it is in classical CJD. So there's more opportunity for it to contact surgical and medical instruments. And of course, as you heard from the HPA study, there are potentially quite a lot of people carrying the infection around. We also know experimentally that it's relatively easy to transmit prions by binding them to metal surfaces. In fact, it's something we use experimentally now. It's a very efficient way of transmitting the disease, and indeed the blood test we've talked about earlier exploits the affinity of prions for metal surfaces as a way of concentrating the infective agent prior to detection. So there's plenty of scientific evidence that this would be an efficient route of transmission. There's plenty of evidence that there are many people out there in the UK incubating the disease, and that the infection would be in tissues which come into contact with surgical instruments. So, but you're right, there isn't so far a documented example of that happening so far with variant CJD.
I'm not quite sure I understood the first part of your answer. Can we tell from epidemiological studies whether or not there is an indication that there has been transmission of variant CJD? I mean, there's no routine testing, but is there any other way that we can have an indication that there has been transmission?
No, I mean, it would be worth speaking to the epidemiologist at the surveillance unit whose responsibility that is, but so far there's been no case that can be readily attributed to prior surgery. But as I say, those are difficult studies because surgery, particularly dental surgery, is so common.
Right. NICE introduced guidelines intended to reduce the risk during surgery in 2006. Do we know if those guidelines have been effective, and are they still relevant at the present time?
I'm not sure I can answer for how that's being implemented in the NHS. That's outside my expertise. I mean, the awareness of this risk some years ago led to a review of standard hospital decontamination procedures and practice. I was a member of the advisory committee SEAC at the time when this was being discussed, and I must say it's one positive thing, if one can say, that came out of the whole BSE crisis, in that did lead to exposure of a lot of bad practice in terms of surgical instrument decontamination. A lot of places audited were found to not be up to best practice, to put it mildly, and a major investment was made, I think of about £500 million at that time, by the Department of Health to bring central sterilisation processes up to standard.
Thank you.
Stephen.
Thank you. Following on from that, perhaps I could turn to you, Dr. Bors. What made DuPont decide to invest in Prion decontamination product in the first place? I know you gave us a very comprehensive introduction at the beginning, but what was the driver?
Well, we saw a need for the product. Also, the fact that there are not prion decontamination products out there that are compatible with medical instruments or delicate instruments. So our product is pH neutral and has corrosion inhibitors, so to help maintain the integrity of the instruments. So we felt that we could fill this gap in the market with a product such as this.
So it was a totally commercial decision?
Well, I would say that there was a need for the product with the recommendations of single-use instruments or tracking of instruments. This could be a more cost-effective addition to the process.
Okay. And so then you, DuPont as a company, invested, developed the product. but obviously it has had some challenges getting to market. Could you just run us through what the barriers were to getting it to the point where in 2007 it was launched, and then what sort of happened in that interim period between 2010 or up to 2010?
Okay, well, if I could give an overview, because I wasn't on the project until 2008, so if I could just give an overview of the main barriers that we found overall. The primary one being that there were no regulatory drivers for the product use. We were told very clearly by the rapid review panels that unless there was such recommendations in place, it was unlikely our product would be taken up widely within the NHS. Also, the fact that it is a pre-soak product that adds an additional step to the decontamination process. It can't be used in the automated washer disinfectors that are the best practice. In addition, Sorry, I thought someone was going to ask a question. In addition, it was very difficult for us to actually obtain approval to trial the product in healthcare settings. We made several attempts to have the product trialled, but we only had one materialised at St Mary's Hospital, and we found this to be a successful trial. But there was a lot of resistance to actually getting the product trialled. We had some practical commercial aspects as well that were a barrier for us. The product has a 6-month shelf life, which is not a problem for the end user, but we have a minimum batch size production run of over 6,000 packs. So without confirmed orders, this would mean that we would be essentially disposing of a large quantity of unused and out-of-date material. And then a further factor was that one of the ingredients in the disinfectant part of the pack was reclassified in December 2010. So it was given a toxic to reproduction classification, which would have meant the whole product would carry this toxic labelling. So this was removed from the formulation and replaced with a non-toxic equivalent, which is sodium percarbonate. And although this was resolved from a formulation point of view, it would have required further validation in terms of the biocidal efficacy tests and the Prion efficacy validation.
So you were developing a product that there had been an identified need for. You developed that product. It hopefully said that or did what it was specified to do, and yet at every stage there were challenges thrown up to almost stop. I mean, were you not working with the end user during that development period and up until the point at which you launched the product, so that it would fulfil more of their identified needs, that barriers wouldn't exist?
Yes, we did. We conducted a large number of interviews with the end user, and there was the feedback that it would be a good product to use, and that I think the main barrier was the fact that it's a pre-soak product, and initially people said this wouldn't be a problem, but obviously further down the line with the need for a validated automated process, this became more of an issue.
Right, okay. So you thought the product might develop into something else at some future point?
There was always the intention that a Generation 2 product would come out of this, but unfortunately didn't materialise.
Okay. And is there any market for the product outside the UK? Did you try that?
We did explore several markets, France in particular, and as well as some other European markets, but we found that the lack of demand didn't really fulfil further investment in this line.
Okay. And have you had any contact with the Department of Health since 2010 when the product was put on hold?
No, we have not.
Do you still have confidence as a company in the product? I mean,
we have confidence that the product sets out exactly what it intended to do. However, it's difficult to see with the feedback we've had from the rapid review panel how it would get implemented within the NHS with the current recommendations. So
for you to be able to restart development of the product, perhaps to make it not a pre-soak but something that could be included in a later stage of decontamination, what would kickstart that reinvestment? What would get the company looking at this again?
Well, the business case would have to meet a set of criteria for product pipeline selection. If it were to be re-engineered to go into automated processes, it would be a complete reformulation, would be essentially starting from scratch, so this would take several years. So there'd need to be significant justification for a business case. Even if we were to proceed with the manual pre-soaked product, there would still be a not insignificant amount of work. It wouldn't just be a case of ordering in the chemicals and putting a product out there. We'd need to have sufficient commitment to volume to justify this. We'd need additional resources from a personnel point of view to support the project. It would be necessary to validate this new carbonate-based formulation product in terms of the biocidal, the prion efficacy and end-of-shelf life data as well. So there would be sort of a number of factors to consider.
I mean, it sounds like it was quite a torturous process to get it to where it was, and I can understand your reluctance to start reinvesting in that. I mean, I think we've heard, and I wanted to come in earlier about when we were talking about the politics of all of this, that if a product fulfills the specified or identified need, is then developed and fills that need, I think someone said it should then be adopted, but it's not because of political, or perhaps for political or financial reasons. Has that market changed over the last 10, 15, 20 years, or has it always been as difficult as it is now to get a product taken up by the NHS in this case? Yes.
Sorry if I can answer that. I would say that the climate that existed around about sort of 2000 to 2005 was one of real concern, and I think the UK blood agencies and the Department of Health were very concerned that there was going to be another not epidemic, but growth of cases of variant CJD by virtue of blood transfusion. And there was, I think, a genuine desire to do something about that. A number of new committees were formed. In the case of the Prion filtration, there was a Prion filter working group formed, of which we were part of, and various blood agencies, not just in the UK actually, but in other European countries as Ireland and France, were also invited onto that panel. So there was a real, I would say, spirit of collaboration at that period, that we're all working together, industry, the NHS, to try and solve this problem. And I think that climate changed. We experienced evidence of that in terms of the PRISM study. You might be aware of this. This was the clinical trial that was performed by the UK Blood Services on the Prion filter. This was a study on 270 transfused units which had undergone pre-infiltration, comparing with 270 units that hadn't undergone pre-infiltration. It was a multi-site study. I think something like 6 hospitals in the UK were involved, and these were major hospitals, not sort of minor local hospitals. That study should have taken, I believe, 18 months to conduct. It took 5 years. One might ask why that study took so long. In fact, SABTO actually commented upon this in 2009. They were concerned that the study was taking too long, but it didn't actually complete until the end of 2012. So from our perspective, that spirit of collaboration that existed in the first half of the last decade sort of disappeared, and we were witnessing an environment where, from our perception, sort of roadblocks were being placed in the way. Things were being stretched and taking longer, and as soon as we had achieved one hurdle, another one is all of a sudden in the way, and that was our experience in the latter part of the development of the product.
Anyone want to add anything?
I think to say something on this about, first of all, there was the financial crisis, and that has certainly made the introduction of new technologies more difficult, not only in the UK but throughout Europe. However, there are certain cases, such as Switzerland, where they've I think 3 or 4 years ago, they had 3 cases of bacterial contamination of their platelets, and they were within a period of 1 year, and because of that contamination of those platelet products, and unfortunately there was a death of a child associated with one of those platelet products, they implemented a pathogen inactivation system because of that. So what can happen, and where this becomes a political issue, is that when it's in the public, when there's public awareness, there is a high emphasis on testing or pathogen inactivation, pathogen reduction of blood products. This is forgotten about after a period of time. And if you combine that with a financial crisis, it means that you see over a period of years a high emphasis, then people forget about it and the emphasis drops down. So that's what we're seeing.
Okay. All right. Thank you, Chairman.
David.
Can I add something there? I spent my whole career as an NHS doctor, and I think it's fair to say the NHS can be quite resistant to change and hard to do new things in it. And with respect to the surgical instrument decontamination project, for instance, I wasn't completely naive about this, and did go and see the then Chief Medical Officer, Sir Liam Donaldson, because we were about to embark, funded by the Department of Health, in a major research programme involving 3 professors and a great deal of resource in my unit to try and solve this problem of how to decontaminate metal surfaces. And I said to Liam, if we do this, is this actually going to be used? Is the NHS actually going to use this? And he said to me, John, if this problem is solved, I'll instruct all hospitals to use it. And I didn't think there was a clearer case than that. But of course, what's happened, one of the changes that have been discussed, what's happened is a change of the relationship between the centre and hospital trusts. And in fact, when we developed this, and actually I went back to see Liam Donaldson again to discuss this, he said to me, well, John, the world's changed, that we don't tell hospitals what to do anymore. This will be a decision taken by individual trusts. And of course, there was no, and he wrote to me in that regard, but of course, then at the level of the individual trust, there is no driver to do this. That they're sat there thinking, well, why should we do this? It involves us changing our procedures, we'll have to change our working rotas, some additional cost, and it simply doesn't happen. So that change in the relationship between the Department of Health and trusts, I think, is also relevant.
Could you tell us when that letter was written? Or when that discussion was taking place?
It's quite a long time ago now. I'd have to go back, and it's probably 7 or 8 years. But I can certainly find the letter if Professor Donaldson's happy for me to send it to you.
Okay, that would be useful. Thank you.
Thank you, Joe. I mean, just following up on that point first. It does seem, I would not describe the military procurement as being an ideal procurement method, but nevertheless, when they identify a need, they then ask people to develop it, and it's produced, and if it works, it's implemented. In the NHS, we have a process of nudges and winks, it seems to me, as far as certainly the commercial sector is concerned, as to what might be useful. But nobody actually takes responsibility for ensuring the testing regimes and the use of potentially valuable technologies within the NHS. Is that a fair characterisation? Professor Collinge, I think probably you're best placed to answer that.
Yes, I think so. I think there are still, it's unfashionable to have central dictates about things, but when it comes to infectious diseases, which don't respect any borders, certainly not the relationship between one trust and another, I think there needs to be a national position on infectious pathogens. I mean, we sort of have this with things like MRSA and C. difficile, which of course have been widely aired in the media and led to a number of actions to make sure all trusts have standards and are inspected as to whether they tick the relevant box, that they've taken the appropriate measures to reduce the risk of those pathogens. But there isn't that with respect to prions, as far as I'm aware. There's no inspection of hospitals, but CQC or anybody else saying, well, have you done a prion risk assessment in your hospital? Are you doing high-risk procedures? Have you taken appropriate precautions? So there's no real driver for things to happen.
We can't run infection control on the basis of whether it's on the front page of the Daily Mail, can we?
No, absolutely not. So I think there needs to be a clear statement. Is this worth doing or not? Are we sufficiently concerned about the risk that we're doing anything? Or we are sufficiently concerned about the risk that we're blighting people's lives by informing them they've been exposed to a contaminated instrument? So presumably we think there is a risk. Then there should be national standards of what we do that all NHS trusts are expected to adhere to.
Yep, thank you. Dr. Burton, I want to ask you about your filtration process. First of all, are you the only company working in this area, or are there other prion filtration processes on the market?
There have been other companies, other products developed by other companies. About the time we started work in this area, a US company called PAL Filtration were also developing their own Prion filter. That fell by the wayside a few years ago in terms of issues with performance, I understand, in the case of what's called endogenous infectivity, the blood form of infectivity. With these filters, it's not really possible to develop them, to undertake the development using variant CJD blood, partly because there simply isn't enough available, because we're processing whole units of blood here. And partly because we need to have some measure of how much infectivity we can bind, and therefore the sort of the brain material tends to be used for that. But there is a difference between the brain material and the blood endogenous material, and the brain material can be relatively highly aggregated and actually relatively easy to remove by normal filtration, just removing small particles. Indeed, it was, in the past people often referred to prion particles because it was believed to be some kind of particulate material. It was later that it became apparent that sort of all blood components are infectious because there's a soluble form of infectivity there, and that's the form that we need to address in the case of red cells and plasma. So developing these devices is not straightforward. It does require fairly meticulous studies conducted over quite a large period of time. But yes, there have been other companies developing these products. Not all of them have proven to be effective with endogenous infectivity, whereas the PCAP filter that we developed has been proven to.
And have any of them been actually used?
I say we, this is Maca Pharma, who is the company that we licensed to manufacture the product. They have made sales of this product in various locations. Actually, Macau province in China uses this material for the local expatriate population donating blood. Otherwise, in Europe, it's really only been used in relation to clinical studies. In terms of normal use, there hasn't really been any usage.
And I think when you were describing it, you were saying it was designed specifically for the architecture of variant CJD protein, that is what it binds to. So does that mean it doesn't deal with other prions?
When we developed our product, we set out to have a broader selectivity as possible for prions. So we screened many millions of different compounds, different types of compounds, over a period of 2 years. And at the end of that process, we'd identified 5 compounds which were able to bind reasonably selectively the normal form of prion, sometimes called PRPC, and the infectious form of prion, sometimes called PRPSC. We looked at different sources of infectious prions. So we looked at BSE, scrapie, hamster, all these different types of materials, and to basically select finally the product that went into the PCAP filter would have a broader selectivity as possible for prions. Now, there is a difference in binding. Like we know, for example, that sheep scrapie does not bind as well as human or hamster or monkey prion. But there is some binding. So it gives a broader level of protection. In the pharmaceutical industry, it's a well-established sort of principle that during manufacture, yes, it's important to test for the presence of viruses, particularly with protein-based drugs, plasma-derived products, for example. But it's also important to have some capacity in the process to bind infectivity if it's present. One feature of a test is that we can only really test for what we know. We can't test for what we don't know. Whereas in a process, if there is some capacity to remove other things, then if a new form of prion comes along in the future, a different animal source, there is some protection inherent in the processing that's been applied that may or may not be picked up by a test.
I think that's really useful. At the risk of having an instant peer review session of this, presumably that would be complementary to any of the other processes which we've had described. If you reduce the potential effective agents within your supply, then you've got less to deal with either by blocking them or testing for them or anything else. Does anyone want to comment?
Well, I mean, we've always viewed testing and filtration as being sort of mutually compatible and desirable
that's
steps
what I was
to take,
thinking.
rather than do you choose one or the other.
Yeah.
If one really wanted a robust protection for blood against variant CJD, one would implement both of these techniques.
Yeah. Does anyone wish to comment? I know the other panelists want to. No. Okay. Thank you very much, Jim.
Thank you, Jan. My question is actually to Dr. Burton, following on from what David said, and also what you said in reply to Stephen earlier. I want to look briefly at the PCAT device and its history. But what I understood in your reply to Stephen was that you felt there was an initial encouragement to pursue such research and underground, but that the rules or the mood changed while you were doing this. And when you said one obstacle was put up after another, I understood that to mean, please correct me if I've misunderstood, that sounded as if there was a degree of artifice involved, that Sabto didn't want to endorse the product and simply looked for reasons for refusing.
I can't comment on Sabto makes his decisions, but I would say that, as I mentioned earlier, that we did sense a change of mood. That what we experienced at the beginning were comments to the effect that, well, if you achieve a CE mark approval for your device, then we could implement it straight away without any additional testing. When we got to that point, we were told, well, actually, we really want to do our own independent testing. So at that point, we had already done safety testing in relation to the impact on red cells, for example, hemocompatibility studies, that sort of thing. We had undertaken our own safety study in human volunteers for the transfused blood, and so it showed there was no issues with that. Studies were underway in Ireland to do exactly the same thing, and the UK Blood Services said, well, we want to do our studies as well to demonstrate that the blood that comes out of the filter really is safe. Initially, Sabto in 2009 made a recommendation that PCAT filtered blood should be made available to individuals born after January 1996, subject to the completion of the PRISM study and a successful outcome to that study. As I mentioned, the PRISM study took much longer to complete.
There were also the efficacy evaluations.
And then, yes, so then later on, the issue of, well, the repeating of efficacy studies came in. Various groups in the UK have been performing these independent studies. There's a study with the HPA, and they undertook a study to look at the binding of endogenous infectivity. That study, we were in confidence. The results were shared with us. We had some issues with some aspects of that study, particularly the fact that the flow rate used was substantially in excess of the maximum recommended flow rate for the device. So there was a flaw there in experimental design. Another study that was undertaken was a sheep study, and as I've already mentioned, we know sheep prions do not bind as well to our device as human or other types of prions. So we don't think that's really a fair and relevant measure of the performance for human blood. So, but those results, I think have been taken into consideration. So, we moved from a position, well, really we could, as long as we gain the legal approval to have a medical device sold and used, which is a CE mark approval, that it wouldn't really require any additional testing. We found ourselves going through successive rounds of additional testing.
So is it your understanding that either A, they couldn't replicate the results that you'd obtained before you submitted it, or B, they applied subsequent and different tests which you never did?
I would say that, well, they were in the ways different tests. I mean, we never performed a study in sheep because we didn't think it was a relevant model for the device. The other study that was done, as I said, there were some differences, and the filter was used outside of its stated specification, basically. So,
sorry, can I just get that a bit clear? Are you saying they tested it for things that it was never meant to do?
They used the product in a way that it wasn't intended to be used.
Right. Okay, so, but you're still supplying, from what you just said, you're still supplying the PCAPs in other parts of the world.
Correct.
Have you abandoned any attempt to get it past the UK?
No, we've not abandoned the product. I think because of the very long developmental period, there is a limit to the appetite of companies to continue investing at a high level in a product if there doesn't seem to be a return on that investment. So I would say that there has been a decline in activity around this particular product. There is work ongoing with another study. This is work that's been undertaken in France by the CEA laboratory, and they have used the PCAP filter to remove infectious prion from macaque monkey blood. This study is subject of some debate right now because the findings are quite interesting and somewhat unexpected. On the face of it, what this study in France shows is that the blood taken from monkeys that show symptoms of prion disease are taken, separated, and then treated in two ways. One, undergoing normal leucofiltration, which is a white cell removal filter step, and then given to healthy monkeys. The other group, through normal leucofiltration, and the PCAP prion filtration, given to another group of normal healthy monkeys. And this study has been undertaken twice, I understand. The results of this show that the PCAP filtered animals are all alive and well today, and this is, I think, 66 months after the original transfusion took place. All of the animals in the non-PCAP filter group are dead, and they died around 30 months of a prion-like disease. And I think this is the debate right now as to what is that prion-like disease, because it's atypical in the sense that the lesions are not really centred in the brain, which is typical of CJD. It's one of the measures that at autopsy tissue histology is undertaken, and if there are spongy holes in the brain, that's very indicative of a CJD disease. Well, that wasn't the case with these animals. There was lesions in the spinal column. If that was a human case, that individual would not be diagnosed as having any kind of CJD. It would be a different diagnosis that would be made. It would be more akin to a disease called amyotrophic lateral sclerosis than CJD. So it raises some issues here, but in terms of the ability of the filter to remove infectivity from a primate model, which is the closest we can do right now to the human situation, it shows in very clear black and white terms that the filter is very effective in removing blood-borne infectivity. It provides a very high level of protection to those in the case of that study.
Okay, thanks, Jeff.
Well, one always comes out of these inquiries a little bit more scared than when we go into them. What pathogen, what emerging pathogen is the greatest potential threat to the United Kingdom blood supply at the moment? Scare us a bit more.
That's not my area of expertise. I talk about Prion risk, but I don't know about other emerging pathogens.
Maybe this is an area that we work on, and in continental Europe today, through climate change, there are growing concerns over new pathogens that are emerging. And in recent years in Italy, we've seen an outbreak of a disease called chikungunya, which is a tropical disease that was brought into Italy in tires, and through a vector called the Aedes albopictus mosquito. And this closed down part of the blood service in Italy for a period of months, whilst to prevent this disease from spreading. And in fact, also in France, there have been cases of dengue fever and chikungunya through transmission of the tiger mosquito, the Aedes albopictus mosquito. And in fact, these mosquitoes have been found as far north as Antwerp in Belgium. And in 2013, these mosquitoes were found again in tires, so they come from the Far East, and also in bamboo. And it's a fairly short trip across the channel for these mosquitoes to come to the UK. As far as I'm aware, they are not in the UK at the moment, but they do carry a number of different diseases. It is also, we've seen that there are have been an increased concern over hepatitis E. In the Netherlands, there was a study done of blood donors in 2013, and it looked at donations of blood, I think, in 2011 and 2012. And of 45,000 blood donors, they found that about 17% of them had at some time come into contact with hepatitis E. Now, hepatitis E, if you're a healthy person, has a small risk. But if you're immunosuppressed, or maybe a pregnant woman receiving blood, or maybe a patient with hematological malignancies, then the hepatitis E can be more of an issue. And in fact, in France, there was a case in 2013 of a blood transfusion of hepatitis E. And now that the French regulatory authorities are looking at this at a much higher light. Now it is the technology that we manufacture is effective in reducing not only the known pathogens but also the emerging pathogens, or some of these more difficult pathogens such as hepatitis E too. So there are many new pathogens that come along, and one of the issues is, can you test for every single one? And the answer to that is probably no. By implementing a pathogen reduction technology, you're able to inactivate not only the known pathogens, to close the window period when used in conjunction with various testing methods such as nucleic acid testing, but also give a level of protection against those emerging or unknown pathogens.
Yes, in your... I'm sorry.
Maybe I just add one comment from a veterinary diagnostic company who has a strong focus on controlling zoonotic pathogens entering the food chain, and that was actually also the case with BSE. What we do see also, and I confirm what my previous speaker just said with hepatitis E, we do see a high prevalence of hepatitis E in pigs, and we do have a test, and there are certain prevalence studies being conducted in Europe. And this is frightening, what we see there, and especially as dietary habits have changed and people are more into eating not fully cooked pork meat, this has a high chance of moving over into humans and potentially could be a hazard to the blood supply.
Thank you. Mr. Talboys, in your written evidence, you say that these emerging pathogens we don't assess them for risk.
Mm-hmm.
Can you expand on that statement?
I think if you have a look in first of all, I think in the UK it really is a very excellent system and that is in place in the UK today, and there is a report, the Serious Hazards of Transfusion report, called the SHOT report, which has shown a very low incidence of transfusion transmitted infections. However, with climate change, it is an unknown aspect, and these new and emerging pathogens appear and can cause issues in the blood supply. And I'll take another example, which is West Nile virus. In West Nile virus, there have been a number of testing that has been undertaken in the UK and seen and there's been a potential increase in West Nile virus. We've seen in the US that in the early 2000s, West Nile virus came into the US, and over a period of years, the incidence dropped down to a low level. I think it was in 2012, 2013, there was again a major increase in the number of incidents of West Nile virus, and this got into the blood supply, and there were increased incidence of receiving this in the blood supply. And I think it's looking at these unknown risks, and it's difficult to risk assess something that is unknown. And we've seen with the weather this year, with the flooding in the UK, there's been a very warm winter again. Can these vectors overwinter and start to bring in some of these tropical diseases that have been unknown in Europe for many, many years? And that's what I see as the potential of how do you risk assess something you don't really know about?
All right, thanks. I've just rewatched, and the band played on. I don't know if you've seen the film. It's really about the United States government's response to the HIV AIDS issue in the early mid-80s. And I suppose the film's a polemic, really, against both the government and the commercial interests in the blood industry, where they were... The people who controlled the blood would not do tests because they were worried it would damage them commercially. The point I'm getting to, really, is that how does our approach differ from other countries? And if other countries are not as good at protecting their blood supply from pathogens, does that have any impact on our blood? Do we import blood from those countries?
Well, first of all, there are some limited importation of plasma into the UK, but the UK is predominantly self-sufficient in blood, particularly red cells and platelets. As I said earlier, the UK has a very, very good record of having safe blood. However, and we're working with SABTO on the report to look at pathogen reduction, pathogen inactivation of platelets today. And this report is predominantly looking at bacterial infection and also the ability to replace gamma radiation from the use of platelets. We would say that there should be a broader look at the safety of the blood supply, not only to look at bacterial infections, which are the largest infections that you see in platelets, but also to extend that to viruses and parasites as well. And this is what we see now that other countries are starting to look at because of the emerging pathogens. If we have a look at technologies across Europe, the technologies for pathogen reduction are used in most countries in Western Europe today, not always on every single blood product, but they are starting to be adopted rapidly. And we are seeing countries like Switzerland, there is a mandated use of pathogen reduction for platelets. We even see that in Africa, that there is now, because of the risk to their blood safety, we're seeing that they're starting to look at these type of technologies. And again, as I pointed out earlier, we have a clinical study looking at the prevention of the transmission of malaria through blood transfusion. The clinical study is actually starting this week in Ghana, and if we're successful on that, we expect to see adoption of our technology in a number of different African countries.
Thanks. My final question, which you partially, if not mainly, answered. What do you hope the current SAPTO working group on pathogen reduction technology will achieve, really? I mean, you've partially answered that. I'd be grateful if you could expand on it.
Well, I think there are a number of steps going through. I think to have a look at the adoption of these type of technologies in the UK to add a further level of protection to the blood supply. And there are still a number of steps to go through. Each blood service has a different way of implementing these technologies. So the next step after the report has been published is to have a look at the technologies and have a look at potential validation of the technologies and see how it fits into the particular process that we have in the different UK services, whether that is in England, Wales, Northern Ireland or Scotland. So I would see that let's wait until the report is published in the next couple of weeks, and then I would see that moving on to a validation of the different technologies and then potential adoption after the validation of the technologies.
Okay. Can I thank the panel for very comprehensive responses to questions this morning.